Welcome back to another episode of Gene Therapy, A Whole New World. It's your girl, Hanna, and today we are diving into one of the most pressing questions surrounding gene therapy. How long does it last? Well, the New England Journal of Medicine recently published an article titled Sustained Clinical Benefits of AAV Gene Therapy in Severe Hemophilia B. And with an increasing body of long-term patient data, we're gaining exciting insights into the durability of gene therapy, especially in hemophilia. Bloomberg even followed up with a piece called Hemophilia Gene Therapy is Shown to Last for Years, featuring our guest today, Dr. Andrew Davidoff. Though before we jump in, here is a note from our sponsor. It's a whole new world gene therapy segment brought to you by CSL Behring and their Hemophilia B gene therapy treatment. To learn more about gene therapy, hear patient stories, and view additional support resources, visit beyondhemb.com or download B Support wherever you get your apps for more information and to manage your treatment journey. So our guest today is Dr. Andrew Davidoff. He is the chair of surgery at St. Jude Children's Research Hospital. And I understand what you might be thinking. Why is a surgeon who works on cancer here to talk about hemophilia? So that's a great question. And here's the scoop. Dr. Davidoff's pioneering work in gene therapy involves using AAVs to deliver treatment for monogenic disorders like hemophilia. His expertise in gene delivery makes him a key voice in understanding where gene therapy is heading. We've heard a lot of buzz about the long-term effect of gene therapy. Now, we're finally seeing the kind of data we used to only hope for. Patients treated over a decade ago are still experiencing benefits. A 2024 study followed hemophilia B patients 13 years after a single dose of gene therapy. And the results were, they were quite remarkable. So let's hear what Dr. Davidoff has to say about the kind of long-term success we are seeing.
I think the data are very encouraging for long-term durability of efficacy of gene transfer for patients with hemophilia. We have just looked at our participants who now have been on average 13 years out from receiving the vector, and their factor 8 and factor 9 expression from the gene transfer vector has been stable for upwards of 13 years. So we're very excited about that.
Wow, that is a huge achievement. 13 years of stable expression from a single treatment. Imagine the impact on a person's life if this kind of durability continues. But here's the thing. Not every patient responds to gene therapy the same way. So, I asked Dr. Davidoff about the variability. What factors shape outcomes for different people?
Not everybody responds to a dose of vector in the same way, and we are trying to understand that to prepare those patients for the different outcomes. Baseline liver disease probably bodes poorly for a better outcome from gene transfer to the liver. But other than that, it's not really so clear why some patients benefit more than others.
And here's another thing that really stood out to me. This long-term durability wasn't something researchers originally expected.
The durability was a little bit unexpected just because nobody had done it yet in humans, been able to follow them for such a long time. Certainly the preclinical studies in animals suggested that the effect should be long lasting, but didn't know until we had human participants in our clinical trial.
So let's shift to hemophilia B versus hemophilia A. I asked Dr. Davidoff, how the outcome compares between the two.
The durability seems to be better for factor 9 for hemophilia B than for factor 8 in hemophilia A, perhaps for a variety of reasons, including the fact that the factor 8 protein is much bigger and more difficult to package the gene encoding its expression, and a little more difficult to synthesize. But other than that, I think long-term results have been quite encouraging.
That's an important takeaway. Hemophilia B is currently showing more consistent long-term results, and the complexity of the factor 8 gene likely plays a role in that. Now, here's a key issue. Gene therapy is currently a one-time shot. The body's immune system develops antibodies to the viral vector, which means redosing is extremely difficult, if not nearly impossible.
When a patient gets an AAV vector, the gene is successfully transferred, but the patient does get an antibody response and maybe even a T cell response to the vector itself that probably precludes them getting a second dose of the same vector. Now, there are differences that can be engineered into the vector that you could get potentially a similar but different in some important aspects vector later on and have successful gene transfer. But that's a little problematic in many ways because the vector is difficult to make, and to try to have a whole sort of shelf of different types of variations is going to be difficult. People are looking into this important question, whether giving perhaps immunosuppression transiently at the time of receiving vector might dampen that reaction to the vector and therefore allow for repeat dosing. But I think probably right now the answer to that question is probably not. But at least for our hemophilia B cohort of patients, it's not an issue because they are stably expressing.
So that's a big deal. For most patients, gene therapy only gets one shot to work. That's why vector design, patient selection, and delivery timing are all so very important. So how do clinicians actually know if gene therapy is still working years later? It starts with factor levels in the blood, yes, but it goes much, much deeper.
So these patients all had severe hemophilia, which by definition is essentially no circulating factor, so that any factor that they now have, which is easily measured by standard means, as long as they haven't recently given themselves recombinant protein for whatever reason. So if that has cleared, whatever is left is coming from the gene transfer vector. So that's a very easy way to generally see that it's still working. We are interested, though, in taking a deeper look at the vector, itself in the liver. And it's interesting, when we first started, the idea that doing liver biopsies in hemophilia patients with a baseline bleeding disorder was really considered outrageous, frankly.
That shift from avoiding liver biopsies to using them to deepen our understanding is a huge sign of how far the field has truly come. So let's take a step back. What makes a gene therapy actually work, not just initially, but for years? Well, it turns out it is all in the design.
But now that we and others have shown that the vector is safe and effective when it comes to bleeding and bleeding after trauma, including, surgical type procedures, that the questions that are being asked about the fate of the vector are so important that now, in fact, it's being encouraged that clinical trials include the opportunity for patients to participate in a biopsy of the liver so that investigators can understand, is it still there? What form does it take? Is it integrating into the host genome? Are there inflammatory cells reacting to it? A variety of questions that can't just be answered by a blood test, that really need to be answered by looking at the target tissue itself. A lot of my research is cancer therapy, and I have been interested in cancer targeted gene therapy. And so I view it, again, sort of the opposite way you phrased the question. I think what I'm learning about our experience with gene therapy for hemophilia, I hope to take some of those lessons and make a better gene therapy approach for cancer and cancer patients.
But there's still a huge hurdle ahead: the cost.
The biggest challenge right now, frankly, as you're probably aware, is the cost to the patients who are not receiving vector on trial, and it frankly has been prohibitive for the most part. It has now been approved and commercialized, yet not very many patients have gotten vector for hemophilia A or B outside of the context of a clinical trial, and that's disappointing. And I would say that's the biggest hurdle right now. We have a good product, but just getting it to the patients in need has been problematic.
That's a tough one, but I'm hopeful that the economics will eventually catch up. Dr. Davidoff, thank you so much for your insights today. This has been incredibly helpful in understanding where gene therapy stands and where it's yet to go. The future is so bright for gene therapy, and the journey we're on right now is just at the beginning. I am beyond excited to see what happens next, and I hope you are too. As always, we will keep you updated on all the latest in the world of gene therapy. It's your girl, Hanna, and thanks for tuning in. And stay curious, stay empowered, and let's keep this conversation going.